Hello, listeners, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and I'm here with Kristin Yarema, Ph.D., CEO at Poseida Therapeutics. Poseida is a clinical stage biotech that leverages its proprietary non-viral gene engineering technologies to create cell and gene therapies. Kristin, welcome to Cell and Gene, the podcast. Thanks for being here.
Thank you so much, Erin. It's great to be here.
Good. All right, I want to jump right in. So me personally and Cell and Gene have worked with Poseida for quite some time, and Poseida's Mark Gergen was a guest on Cell and Gene, the podcast in 2022. And at the time, among other things, he and I discussed why the company is moving from AAV delivery in favor of nanoparticle delivery. And so I encourage everyone to go back and to listen to that. It was a great episode. I want to talk to you about, what's your focus for 2024?
Absolutely. Yeah. So, Poseida's focus in cell therapy is on a fully, purely allogeneic approach. The company actually started out working on autologous CAR T cell therapies, but very rapidly pivoted to a fully allogeneic approach. And our technology is quite unique. So we're at a great point heading into 2024 because we now have 3 clinical stage programs. So it's a real portfolio. 2 of those clinical stage programs are in hematological malignancies, and the third is in solid tumors. And so, to be very honest, Erin, it's about executing. It's about executing and advancing that pipeline in clinical development. And as you'll appreciate, in oncology and in cell therapy, there is the potential for, studies to move quite rapidly. And then also that executional focus extends into our manufacturing arena as well. We have our own clinical manufacturing facility. We do all our own clinical manufacturing for all our own programs. And so, we'll continue to put up batches and develop our process science and technology there too. So it's really kind of both sides of what you need for successful cell therapy. And, I'm a people person. I think people are important. It's one of the things that drew me to Poseida. So, it's going to be really important to me that we make all that progress while maintaining the really very special culture that Poseida has. We have really passionate people who care deeply about patients, who care deeply about science and new technology. And, and it's a great place to be. We were recently named number 2 in the small employer category for the Biospace 2024 Great Places to Work. And so, it's really, I think it's a great foundation to build upon and carry all of that forward.
Excellent. Well, and congratulations on the award for Best Places to Work. That's to your point, people are the most important part of what we're doing. So that's great. So you mentioned Allo, and that's kind of where I want to go next. From a cell therapy perspective, your clinical and preclinical candidates are designed as off-the-shelf treatments for cancer. So I want to dig a little bit more into the why behind that. And then if you could talk us through your pipeline.
Absolutely. Yeah. So, at Poseida, like I said, we are really all team Allo. And, if anything, we're on a mission to demonstrate what we really believe to be true, which is that allogeneic T cell therapies can be the equal or better relative to autologous T cell therapies. So, why do we think there's a need for Allo? Why the focus on Allo? I'd be the first to admit Allo's been a little bit out of vogue lately as the autologous CAR Ts have put up, demonstrably excellent data. But, you really have to think about, the time when all these therapies are in the marketplace and what are customers really looking for and how do we best meet their needs? And I like to think about, the healthcare system is complex. I like to think about patients, physicians, and payers all as groups of customers who are very important. And, to really meet the need of those groups for large numbers of patients, right? I mean, we believe that the day can and will come that cell therapies should be used, relatively early in the course of cancer treatment. Well, if you think about a disease like multiple myeloma in the second line, just in the US and, 5 largest countries in Europe, you could be talking about 20,000 patients per year. Lymphoma is probably similar. So you have to have a therapy that can be delivered to meet the demand of that number of patients. And you need to meet the patients where they are. That means you have to be able to, distribute it to lots and lots of different centers. It has to be easy to use. It should be available on demand so that, when the physician is having that conversation with the patient in front of them, he or she can say, I think I know what's the right choice for your line of therapy here. We can get you started on that in a day or a couple of days, right? And it's easy. It doesn't involve an invasive procedure like apheresis. It doesn't involve waiting time. And if we think about the payers as a customer group, and I always do that. Payers are not adversaries. They're not, they are another important customer group in the healthcare system. And, value in medicine is increasingly important. It's always been important. That continues to be a focus. And it's just very expensive and will continue to be very expensive to produce autologous cell therapies, even if people make improvements in their capacity and in their manufacturing processes. We believe that we have the ability, even today, to produce our therapies at a cost of, $10,000 per dose or less. We can get 100 doses, roughly from a single batch, from a single patient donation. So, we can offer the volume, or, someday should be able to offer the volume of product that really meets people's needs, so that they can make that therapeutic choice in the moment. And, that there's a lot of flexibility to offer product at a reasonable cost that will enable patient access. And I just don't really see how autologous cell therapy is ever going to be able to bring all of those factors together very easily. I just don't see it. It was very interesting at this year's ASCO. It was really the first time that I had heard the dialogue in cell therapy. And that's where we saw some of the very best, autologous cell therapy come out in early lines, like, second line in lymphoma, multiple myeloma for autologous cell therapies. But right after, in the scientific sessions, right after the data was presented, that would be followed by presentations from clinicians talking about their real world study, experience at their site and the difficulties they were having in, meeting the needs of their patients, even at the volumes, in very late line disease where those therapies are approved today. So if you think about those challenges and you play them forward to the day when you want to treat large groups of patients in relatively early line disease, you just need an altogether more scalable approach. So with regard to our pipeline, Poseida's unique approach is really quite unique. We are all about the T stem cell memory cell. So when we think about CAR T, we would say CAR T is not CAR T is not CAR T. So T cells, there's a whole spectrum of different degrees of differentiation of the T cell. And while the cells that actually kill the tumor are quite differentiated and kind of at one end of the spectrum, we've really chosen to focus on the most naive type of T cells that that exist, which are the naive and the T stem cell memory T cells. Well, those products that have, stemness and particularly stem cell memory have really been shown to correlate the best with clinical responses. So they're the kind of cell we believe that you want, but they're very difficult to work with. They're quite rare in the blood to begin with. They're difficult to put genes into. They're difficult to gene edit. They're difficult to manufacture. And so Poseida has a completely different approach with technology where we don't use the viral vectors and viral technologies that most companies use. We use transposons and a unique gene editing system that we've developed ourselves called Cas-CLOVER. And those two things work together to insert genes into T stem cell memory cells and to preserve that stem cell memory phenotype all the way through our production and manufacturing process. So we end up with just a very, very different product. It's very, very rich in T stem cell memory cells and central memory cells, and very unlike the other products. So we have 3 programs in the clinic. Our lead program with data at ASH 2023 is a program called P-BCMA Allo-1. It's a fully allogeneic BCMA-directed CAR T. Also, in the heme space, we have P-CD19, CD20 Allo-1. That's a first dual allo CAR T for B-cell malignancies. And then in solid tumors, we have a program against a target called MUC1C, mucin-1C, another allogeneic CAR T. And all 3 of those are in phase 1.
Excellent. Thank you so much. And that's actually what I want to talk about next, with regards specifically to the FDA clearance for your IND on CD19, CD20 Allo-1. And that's the dual targeted allogeneic program that is expected to report initial data in 2024. Is that right?
That's correct. Yes.
Good. Okay. I was really hoping you could provide some updates that you can share on the progress, even just right now.
Yeah, absolutely. So just like you said, Erin, we cleared the IND recently, and we feel this is a very exciting program. It's the first fully allo dual CD19, CD20 CAR T that we know of, right? And we're excited because it carries 2 full-length CARs. So that's one of the beauties of that, unique transposon approach that I mentioned. We can put in, a large number of genes. So it's not 2 tandem binders. It's not a mix of cells, some carrying one CAR, some carrying another. Both full-length CARs are present in a single cell. And having a product that can target both of those antigens, we feel is very important because it's well known that antigen escape or loss of the CD19 antigen in particular is a real problem with the CAR Ts that we have today. So this is a product that could potentially be used in patients that have either one or both of those antigens, CD19 and CD20. So we've been activating sites. That's going extremely well. There's a lot of interest in the study. And our first patient dosing is happening very early in 2024. So we should be on track for data in later in 2024, just like you said.
Oh, that's wonderful. We're looking forward to hearing how wonderful it's doing when the time comes. So that's good. I want to talk a little bit about, more broadly, kind of a sector at large discussion, if you will, about some of the biggest hurdles that the cell and gene therapy space faces when it comes to the development of off-the-shelf therapies. And so, one, I wanted to get your take on that. Like I said, some of those biggest challenges, what are they? And then how might 2024 see some real progress to maybe addressing, maybe overcoming is a bit strong, but addressing those challenges?
Yeah, I love this question. So, like I said, there has been a kind of a recent skepticism about allogeneic cell therapy. That wasn't originally the case. People were very hot on allo. We need allo for all the reasons that I mentioned. And then the field kind of hit some speed bumps. Some of the early companies working on allo, faced some challenges. And now, there's been, I would say, more of a pendulum shift back to auto. So in a way, I would say that one of the biggest hurdles, but also the biggest opportunity, is just that no one has been successful yet in putting forth a batch of data from an allogeneic CAR T program that really looks like it, stacks up well against what the auto CAR Ts can do, or for that matter, bispecifics would be another category where you'd want to think about some comparisons. So I do believe that, we're approaching a tipping point, an inflection point around that. And I can talk about that. But why, right? It begs the question, why? Why hasn't anybody been able to demonstrably crack this yet? And I think we at Poseida would say that's because you need an answer not just for, one aspect or a couple aspects of the problem. You really have to have an answer, a technology to do 4 key things. So first, you really have to start with the right cell type. And I talked already about the T stem cell memory cells, and that they can be very hard to work with. The technologies that people have been using in the past that are viral based, they don't have the capacity that we do with the transposon, but they also have the effects of pushing the cells further down the differentiation path just by working with the vector as you try to, put genes in or if you're gene editing things out, you have to activate the T cells. That tends to differentiate them. So getting a high enough T stem cell memory product, I think is the first challenge. But then you also have to have a way to put your CAR genes into that right cell type. Again, we really think a non-viral approach is the way to do it. And we know that our system piggyback prefers to transfect genes into naive and T stem cell memory cells. You also it's allo, so you have to think about gene editing, right? So you need a system that can do that with high fidelity and while preserving the stemness. So we take out the endogenous TCR. We also edit beta 2 microglobulin. So you need to be able to do that, showing safety, right? Showing that you don't get graft versus host, but also host versus graft, right? So your gene editing has to be dialed in a way that you have enough of a window that your cells can get in and do what they're supposed to do, which is expand in the patient, traffic to the right type of tissue where the tumor actually is, differentiate into the right type of cell, that effector killing cell type, and then, actually lyse the tumor. So getting that editing correct is the third challenge. And then lastly, you have manufacturing. You have to be able to manufacture these allo T cells at scale, which is also very challenging. We have a third proprietary technology that we call the booster molecule, which means we don't have to add any cytokines or additives. But if we use our booster molecule, we can really get the T cells to grow very well in culture without differentiating. We purify them, and then they can be, frozen and stored, ready in inventory to ship to, the patient right now in our clinical trials. So you really need the full package, an answer for every one of those problems. And, it's hard to put together that set of capabilities. There's some other challenges as well. I could talk about the challenges all day, but I do think that we are going to see companies start to deliver data that shows that allogeneic therapies can have efficacy profiles that are on par with autologous CAR Ts, and may in fact have the same or even better safety profiles relative to the autos. So I think it's an exciting time.
Yeah, agreed. Okay, so back in August, in 2023, there was a $25 million strategic investment from Astellas in Poseida. And I was hoping you could talk to us a little bit about how that investment has been able to help you move the needle on clinical data.
Yeah, thank you, Erin. Yeah, so we're very very excited about that investment, and yes, so Astellas has invested in Poseida. They're keeping close watch on us. They have a board observer seat. They have a right of first negotiation on our solid tumor program, the MUC1 program, for a period of time. And, I think it's a great illustration. We have, I hope I've convinced you that we have a lot of technology. And with all of that technology, we've got a lot of optionality, right? There's so many things that we can do. And so being able to get investment from strategic companies, strategic investors like large pharma companies, is just fantastic, because it enables us to fund things, and continue to grow, even though the capital markets are incredibly challenging. And that's a model that we love. We've used it heavily. We will continue to do that. In fact, we really, over the past few years, have had either investments or major partnerships with 4 big pharma companies. So Novartis led the initial Series C financing for the company, and they're still a big stakeholder. On the gene therapy side, we had a big partnership with Takeda. Now, Takeda chose to just exit gene therapy, so those programs came back to us, but their support helped us advance them, and that's very important. And now, we will find the right next steps for those programs. And then probably our biggest partnership is actually with Roche, which is a very, very extensive partnership. So both the BCMA and the CD19, CD20 CAR T programs are part of that collaboration. They are licensed to Roche, but the collaboration is bigger. It covers a number of hematological malignancies. So there are a couple other programs that they have an option on, and there's a very large research collaboration component too. So, I think these things are really, beneficial. Our partners get to watch us. We benefit from the funding. They learn a lot about what we're doing, and also have, advice to give us, a lot of disease area expertise, et cetera. So, for us, it's a fantastic model, and we feel very fortunate to be in that position.
Good. Okay. Thanks for that explanation. That's really helpful. I asked this same question of Mark, during my conversation with him in our episode, like I said, from 2022, and I thought it would be really a great question to ask you two to get your perspective. Since Poseida is a company that develops therapies across the board, like we've discussed here today, what has been your biggest takeaway to ensure or work toward scale up? Yeah.
So manufacturing scale up is central to developing cell therapy. And any company that is, taking the mindset, well, we're going to double down on clinical development and kind of see what we get, and then we'll get around to manufacturing later, I think is I believe that's a short-sighted approach. So we've been very, very mindful and very planful to work on a single platform type process. So all of our therapies are using the same platform type process, which, as I said, we're doing in our very own clinical manufacturing facility. So we're not using CDMO support either. And what that means is the learnings that we get, it's very easy to apply them and, very rapidly across all of the programs. And then you have that elegance of getting something worked out that can be used broadly. So think about your INDs, for example, right? We've had great success getting INDs cleared, quite quickly and with little trouble. And a lot of that is our ability to reference, what we have been doing in our earlier programs, and to point out that it's entirely similar. So I think that's really important, and that's kind of a how you do it. But beyond that, I would come back to people again, right? So this is where the teamwork aspect, and especially cross-functional, collaborative, high-performing, agile style teams, all of that is just key. And that's, we might usually think about that being between research analytical to process development, and then maybe process development to, larger scale manufacturing and quality. And that's absolutely true. Those interfaces need to be, very, very strong, very collaborative and very tight. But I would say it extends beyond that. It really takes the entire cross-functional team. So, for example, our clinical development and clinical operations people have been outstanding at providing the right physician and customer perspectives back to process development and manufacturing, so that we don't build some aspect of our process in now that would make for a more difficult customer experience when the therapy is actually commercialized. So you really want to think end to end, and you've got to have people that work well together to do that. So.
For sure. And thank you for that detailed explanation in it. And like you mentioned earlier at the top of our call, that people are central to all of this, in addition to the end-to-end functionality. So makes perfect sense. And thank you for the detailed explanation, like I said. All right. Well, we are at the end of our formal discussion. And so at the end of each episode, I like to talk to my guests about who they are when they're not developing and manufacturing and commercializing life-saving therapies. And so my question for you is, well, I guess first I'll state that, regarding, New Year's resolutions as we kind of come into 2024, I'm not a big New Year's resolutions fan, but I kind of believe in continuous self-improvement. And so I was wondering, what are your intentions, plans, what have you, for 2024 from just kind of like personal fulfillment and just fun aspect?
Yeah, absolutely. Well, yeah, so I would say I'm also not a big New Year's resolution person, but I think, well, why New Year's, right? So why should be anytime.
Sure.
So I guess for 2024, I joined Poseida in this late spring of 2023. And I really, don't know the San Diego area, that well. It's an incredibly vibrant biotech community and really exciting to be a part of. It's just a fun place and a fun, exciting center of innovation for biotech. But it's also just a really, really nice area.
Yes, it is.
Beautiful hills, ocean, beaches, and people with a really good, kind of work hard, play hard, sort of mentality. So I think for me and my family, one of the things that we really want to do in 2024 is, get to know the area much better and really, I love to cycle. Back in graduate school, I was actually on the cycling team, but LA is not the best place for that because there's too much traffic. LA is you know, where I have been based, you know, in at earlier roles in other companies. So, you know, the ability to get out, do some of those outdoor sports. And I have two daughters, 13 and 11, and they are just crazy, you know, mad about animals. And so, you know, places like the San Diego Zoo and Safari Park, I'm sure, you know, are going to get heavy traffic from our family. So really looking forward to that.
Oh, that's so exciting. Well, I wish you well as you and your family get to know the San Diego area better. I'm on the East Coast, and whenever there is an industry conference in the San Diego area, I'm so excited because I just love that area. I've only ever visited professionally for conferences through the course of my career, and I absolutely love it. So I'm slightly envious that you get to explore from a personal perspective too. That's great.
Well, if you're ever in San Diego, you know, you should drop by and see us, Erin. So.
I absolutely will. My children are exactly the same ages as yours. So literally to the exact ages. So, so I'll bring them along too.
Fantastic.
Yeah. All right, listeners. Well, that's it for this episode featuring Poseida's Kristin Yarema. Kristin, thank you so much for your time today and for all the valuable information you've shared. And we look forward to talking to you and seeing what Poseida's up to all throughout 2024.
Thanks so much, Erin. It's been great to be here.
Absolutely. And listeners, we hope you enjoyed this episode as much as we did. Visit cellandgene.com for all the content you need to perform your role even better. And we'll talk to you soon.